Okay, so we've heard many different ways of thinking about interactive roles between the microbiome and environmental health, certainly mediating fate and transport, which is a clear and obvious issue because of the role of host microbiomes in metabolism and toxicokinetics, that the host microbiome itself and microbiomes in the environment may be targets for environmental toxicants. Antimicrobials could be considered perhaps efficiently as environmental contaminants. I want to stress something new that we haven't talked too much about in this interaction between the environment and the microbiomes, and that is horizontal gene transfer. But I also want to re-emphasize the real importance and excitement of Dr. Vijay Kumar's presentation, which is that when you modify the host microbiome, there may be very important and striking changes in risks and expression and severity of acute and chronic disease. So we've kind of let you put that data out there, and we didn't really deal with it, but I want you to know that I think it is highly important. So this question of sort of mutuality or mutualism with a cost kind of is a way of linking these concepts. And I think if I had to sum up the four things that really struck me from being here, and there were many, many of them, is the first one is that it's all evolution, stupid, to use a phrase from inside the Beltway. You really want to understand what's going on here, you've got to go back and remember evolution. That might not fly so well outside the Beltway, you know? Right, I'm sorry, but among friends here. The notion of the microbiome being the 11th organ, this whole notion of mutualism with a cost, which is really interesting. I told Les that I was struggling with this concept, being informed only by Wikipedia, as to whether it could be imported into this discussion. I hadn't really thought of it as with a cost entailed. And then the notion that's also been discussed here, developmental microbiomics, and that links, of course, to a major domain of environmental health concerns and public health. We did finally discuss this morning, I think, a bit more, the notion of incorporating the ecological perspective, that it's not just the landscape, it's not just the physical landscape out there, but the whole landscape of our bodies. And the fact that I think Carl just alluded to, that even within the gut, there's a complex landscape that we need to consider. So there's a lot of useful thinking we could import from that domain. And you know, we really choose our words from where we start, and I think Vince gave a very nice paradigm of that, you know, kind of confessing, how do we get into this to begin with? So I really got into it from the point of view and the concern about antimicrobial resistance, which, like so much in the field of microbiology, we now recognize is not just a simple problem of like good drugs, drugs we're concerned about, drugs of high priority, of clinical importance, et cetera, and bad bugs, that is, the bugs we've recognized as being associated with diseases. It might not even be a problem just of the microbiome, and that's one topic I'd like to raise to us, and suggesting one of the themes that I came here with and I'm taking away with me is that maybe studying the microbiome as a collection of organisms might not be enough to inform us about everything we want to know about positioning the microbial world as part of us and as a mediator of things that get to us, and also something that's out there in the environment. And I see it, therefore, as the problem of locating the resistome. The resistome has been defined by Gordon Wright as others as that collection, that resource base which the microbial world can access in order to respond to stresses. And those are largely, but not, of course, completely, defined by access to genes that encode for resistance to those stresses, be they metals, be they antibiotics, and of course there's a range of other stresses. And then thinking of that in the evolutionary context, I've been well schooled here to remind myself of this in both the long view, but possibly also, may I say, in a crisis view. And I think that was just raised by Ann Summers, that we might be actually in a somewhat shorter or speeded up phase of microbial evolution and our interactions with microbes. So influential experiences, these are mine. And that is, you know, Vince was influenced by the hospital and the demands and challenge of treating infectious disease. Margaret and others were challenged by their studies in evolutionary biology. This is what challenged me, going into a modern American chicken house. And this can only be described as a really badly run hospital. You have the patients or as I tell my friends at Hopkins, you're actually a pretty well-run chicken house here. Somehow they don't like that. But you have the patients all crowded in here. They are actually going to be walking on top of their shit for their lifespan. Others come in and out of this at will, flies, rats, humans, other birds in this case. Sometimes they're even open, in the open air. And so I think we should really think about this in the way that we have thought about the clinic or the hospital as a driver for microbial evolution. But this is something that really goes far beyond anything that happens in clinical medicine. I'd also say, you know, if you adopt the Willie Sutton principle that you want to know where the crimes are happening, you look to where the money is. So I would suggest to you, if you're thinking about the laboratory of microbial evolution and the contribution of the stressors of antibiotics, you need to know where the antibiotics are. Now, we've had a lot of debate about this, but these are actual data that resulted from a data call-in by Dr. Josh Sharfstein when he was one of the deputy administrators at the FDA. So these are at last the real data. These come from the industry. And what these indicate is that 80% of total antimicrobial production in the United States goes into feed additives for livestock and poultry grown for human consumption. 80%. Every single major mechanistic class of antibiotics has been permitted for this use. FDA, are you listening? Because that's who permits it. So this, if you really want to know where the driver is, and some models of the evolution of antibiotic resistance are essentially engineering models of pressure, this is where it is. And coupled with those conditions of ready transmission and lack of infection control. So, you know, when you go to the farm, these are some data from the USDA, from Crayford Orca's lab down in Georgia, and just shows, let's look, this is just representative of sampling that's been done by that lab over a 2-year period, but you can carry it forward, and what you can see is when you start to isolate, for example, E. coli as a sort of reservoir species, if you will, from the gut or from the feces of domestic animals, you can see extraordinarily complex patterns of multi-drug resistance. This is where it's happening. It's in this environment. And so if you go into the chicken microbiome of the chicken gut, you see an extraordinary distribution now of resistance phenotypes. And this leads me to think that maybe characterizing the microbiome by the species may not be fully informative. Maybe we need to start looking at genetic elements and how those may change, even though the taxonomy of this milieu is unchanged. Because certainly for the purposes of understanding the risks of antibiotic resistance, this is what I want to know. I don't want to know if there's more Campylobacter in chickens. It would be hard to have more Campylobacter, but I really would be very concerned if there were plasmids encoding multi-drug resistance now being transferred into Campylobacter. So it just makes it even more complicated, I suppose. And this is a major, you know, led us to start thinking of this problem, not one of bad bugs, good drugs, but really starting to think about the resistome. And this is just representative of a lot of thinking that I know Anne has done, and Abigail Sawyers and others, on the resistome and the importance of this particular activity that we've been involved in, that is raising animals with antibiotics, showing the contributions and increases in really transferable genetic elements into the soil from the disposal of waste from chicken houses. And I've started to think actually about the resistome as cloud computing. It's very hard, you know, don't you think, Vince, not to admire microbes? I mean, they are so far ahead. So they had cloud computing long before Bill Gates or Google, because that's really, I think it's useful to think that that's what the resistome really is. This is this resource of all this genetic knowledge, which can be anywhere within the microbiome. It can even be stored as naked DNA in soil. And microbes have very efficient means of obtaining that and then transferring it back to their perspectives and then within small communities of microbes within microbiomes. And this is really why resistance spreads so quickly and why it is, this becomes, I think, demands upon us to go beyond the microbiome and start asking what's happening within organisms, not simply enumerating organisms, though that's by itself an enormous task. Moreover, it's not only cloud computing, it's really Facebook cloud computing. So once again, maybe they could challenge you know, the patents on Facebook. But this is an extraordinary paper which suggests that there are actually genetic exchange communities that are constructed within a microbiome. So it's not just kind of random or stochastic or Markov chain movement of genetic elements, but they actually set up preferential patterns of gene exchange, which these authors describe as cliques. Sort of reminds me of my teenage daughter and her friends. But you know, not all microbes are equal. There are cliques and frenemies. I like your use of that term, Vince. I mean, this is it's hard not to think of purposefulness going on here, but this is an extremely efficient way to have access to an immediate ability to exchange information. So these are maybe the tweets in the microbial world. And you know, one other point. A point was made that this kind of network doesn't necessarily have to be in close physical proximity. And it was suggested, I think Margaret did, that shared ecologies might promote these sort of links of genetic exchange among and between microbes. But I do want to remind you that geography is not really a very big barrier for bacteria. We move around, we send animals around, wastewater moves around, but even the natural world moves bacteria around. This is an extraordinary mapping that was done by Griffin, showing the patterns, global patterns of movement of dust. And in a separate paper, someone showed that these dusts carry viable bacteria. So their idea of geography is quite a bit different from our idea of what is proximate and proximal. In fact, one of my students is now doing some studies in the West Indies tracking resistant bacteria from Africa. So this is very real and very effective. So it leads me to wonder if we're living in a new stage of animal-human microbial evolution. I guess it's maybe good that Margaret isn't here, because she would probably call me to task for this. But I think we are living at a time after the massive deployment of natural products as antimicrobial agents. Both of those are important. Most of them are natural molecules, so the microbes have seen them. They have the knowledge. It's packed in their resistome. But we have concentrated these, and we have deployed them in a wide range of uses, the majority of which, for example, I would argue in this country are inappropriate, unrelated to the control of animal or human disease. There has been a rapid increase in plasmids. People who have done sampling with cores and other kinds of analyses of the history of microbial diversity, one of the striking things is really the increment in plasmids, which is the packaging for these mobile genetic elements. And moreover, they have become more complex. I just showed you that increasing complexity in terms of genes packaged in a cassette taken out of the farm environment. But generally, they have become more complex, and this, of course, contributes to the expansion of the resistome. So it's not single resistance genes at a time, it's packages of them. And I would suggest possibly that now we're living in an age of increased importance of horizontal gene transfer as a driver of microbial evolution. So not simply pressures selection among colonies of organisms, but drivers of what are genetically distinct and novel organisms because of the ability of the incorporation of external DNA into the backbone of a defined strain or species. So these then, therefore, to recapitulate, all these various aspects make this a domain of interest and appropriateness of interest from NIEHS. And so I thought, as a thought experiment, how do we do this? Do we have an example where we would show how we might put the microbiome together with kind of a contaminant we think a lot about, or at least I think a lot about? And the example I came up with was mercury, because I thought it had elements that suggested where incorporating the microbiome might really amplify and possibly even change the way we think about mercury. So it's just meant as an example. It's a major environmental risk, as many of you know. It's also quite clear, and we've known for perhaps 60 years, that the environmental fate and transport of mercury is regulated by microbial metabolism, and the whole ecological food web transfers that result in bioconcentration and bioaccumulation are dependent on microbial action, and human toxicity varies with these kinds of events that involve microbiome processing out there in the external environment. That we've already known about. And so you know that mercury is biotransformed in ecosystems by bacteria. Methylated mercury, which is the product of that microbial action, is more readily bioaccumulated, and it's one of the most widespread environmental toxicants, both in exposures and health impacts. This is just the mercury cycle in the environment, but I want to stress that it involves both methylation and demethylation, going to some of the points that Tom raised. We generally think about this as a methylation pattern, that it takes inorganic mercury coming from runoff, surface runoff, or atmospheric deposition, turns it into methyl mercury, and that's what's taken up by biota and incorporated along a very classic food chain analysis. You may not know that mercury is also biotransformed by the gut microbiome, and Tom and others have indicated this. And this can both take inorganic mercury and methylate it, and it can demethylate methyl mercury. This challenges a great deal of our exposure assessments about mercury today. It's also important, I need to remind some of my friends, but even at EPA, that inorganic mercury is also very toxic. So taking methyl mercury and turning it into inorganic mercury is not a pathway of detoxification in the environment or within ourselves. Moreover, inorganic mercury is quite interesting because it affects the gut microbiome through the mucosal immune system. We and others have been looking at the specific effects of inorganic mercury compounds on mast cells. And mercury affects the environmental microbiome in another way by driving antimicrobial resistance through horizontal gene transfer. So it kind of has aspects that could possibly be elucidated and enlightened by thinking about the microbiome. So I indicated that inorganic mercury is also toxic, and in one specific way, it's clearly more toxic than methyl mercury related to immunotoxicology. It's more potent at inducing autoimmune biomarkers and phenotypes in mice, at inhibiting host response to malaria in a murine model, increasing circulating autoantibodies in humans, altering cytokine production by cells taken from humans, and this is a variety of work that we and others have done. I'm just going to skip forward to show you some of the human data. This is looking at people who are exposed to inorganic mercury as opposed to people who eat fish. And we're looking at two biomarkers of immune alteration related to autoimmunity, which is one of the cardinal effects of mercury on the immune system. One is anti-nuclear antibodies and antinucleolar antibodies. What I just want you to take away from here are the differing appearances of these four pie charts. Over here we have a reference population also taken from Brazil, where this use of mercury is predominant. And I think you can see that these different colors relate to different tighter concentrations at which you can detect the autoantibody. And that in the persons exposed to inorganic mercury, the concentrations go up considerably higher, such that you see 25% of the population expressing detectable titers at a dilution of 1 in 160, which is extremely high. And similarly for antinucleolar antibodies. This is also dose-related in terms of looking at exposures to mercury. In contrast, this is a population with very high exposures to methyl mercury through fish. You don't see much change at all in anti-nuclear antibodies as compared to a referent population. You do see some increment here in the antinucleolar antibodies related to the levels of methyl mercury, but it's nowhere near as potent as the effects of inorganic mercury. So this is really a very potent material. And I also want to point out that there's a great deal of exposure to inorganic mercury through this use in gold mining throughout the world. And very high levels have been detected. And it's possible that the rest of us may be internally exposed to inorganic mercury much more than we've ever calculated because of demethylation in the gut. And you know, if you just measure fish intake and measure hair mercury, you're measuring the cycling of methyl mercury. If you want to know what happens to the methyl mercury from the fish you eat, you have to do a much more comprehensive analysis of internal biomarkers of mercury dose. Mercury also affects the gut microbiome, probably through actions on bacteria, which Anne and others have discussed, but also on the mucosal immune system. We and others have shown that inorganic mercury is a very powerful agent at disrupting both TLR4 and TLR5 signaling, macrophages, and their representation in the central nervous system of microglia. So it can, this notion of the microbiome being a target for mercury within organisms has not been explored. It also is interesting because it participates in antimicrobial resistance, and it does this through two mechanisms. related to cross-resistance and co-selection. So metals and antibiotics can share mechanisms by which microbes deal with pressures from one or the other. This is primarily through shared efflux pump mechanisms. And thus, when you have exposure to a metal, be it copper, zinc, arsenic, I think silver as well, from what Anne mentioned yesterday, and mercury, you engage these efflux pumps, which at the same time can pump out a range of antibiotics. So that's a shared mechanism. For the purposes of metals, it's an important mechanism. For the purposes of antibiotics, it results in a fairly low or mid-level range of resistance. However, there is another mechanism, and that has to do with co-selection. Mercury resistance genes, that is the separate genes for both inorganic and methylmercury, can in fact be located on transmissible genetic elements which also contain genes for antibiotic resistance. And this is just a representation of one of these. Here's one of the mercury resistance genes. Another one here, you see also tetracycline resistance, and this has been described now for a number of other resistance genes. The important point about these complex resistance transposons or transmissible elements is that any one of these stressors can drive the movement of the element. That is, its transfer within a microbiome. So let's say that at this point you are concerned about preserving resistance to ciprofloxacin. You want to prevent that from happening. If there is a MER gene there, exposure to mercury will generate the movement of that cassette, which will also propagate ciprofloxacin resistance. And in fact, we have some epidemiologic data that suggests this can happen. This is very rough data, but this is the kind of study that's extremely challenging to think about how we might do in reality. And this is a very interesting kind of experiment of nature done in French Guyana. The thing about French Guyana was there were areas where people were using mercury for this purpose of recovering gold, particularly in the areas where some of the indigenous tribes live, the Wayampi here. So they had high exposures to mercury. And then they studied a group of Europeans who lived mainly in the city, had very low mercury exposures. This is described sort of ecologically, but they also did the measurements to prove this. But they selected a group of Europeans that reported recent clinical use of antibiotics. And so what you saw here is that in the population that had very low use of, and no current use of antibiotics, if you looked at E. coli taken from stool samples from these populations, they had very high carriage of MER-A genes for inorganic mercury and antimicrobial resistance. So you're seeing a kind of co-selection through this process, probably of horizontal gene transfer. They did look at transposons as well. So do we really now need to think about mapping metal contamination and antibiotic pressure to understand resistance in the landscape? This was suggested by Singer. He was looking at copper and antibiotic use in agriculture, but it certainly points to the importance of considering both of them within environments. And in fact, Jose Martinez from Madrid has actually proposed that both antibiotics and antibiotic resistance genes should be considered environmental pollutants. And I guess we've heard that maybe EPA thinks that antibiotics might be an environmental pollutant, but it's beyond the scope of EPA's authority to think about antibiotic resistance genes. And Martinez points out that antibiotics are mostly short-lived in the environment, but under conditions of, say, using animal waste might be continuously added, so you'd always find them. But antibiotic resistance genes are considerably more stable. He also makes the very interesting suggestion that we might even think of them as being bioaccumulated in the way we traditionally think about the way pollutants move through environments. When they get incorporated into microbiomes, and they will be persistent within that microbiome. That's his little hot spot up there. And this, I think, is probably enhanced by two things. One is the realization that, in fact, the so-called cost of resistance, the notion that a microbe takes on a resistance phenotype at some cost, metabolic, reproductive, et cetera. But as Levin and others have shown, that in fact it might be equally strategic for that microbe to acquire or maintain an additional mutation which allows the cost of resistance to be abated. And this might be particularly advantageous within a microbiome when the microbiome itself, as an entire community, is existing in what I would call a human-made sea of antimicrobial pressures. It's not like the laboratory where we add antibiotics and we take them away. In many of these environments, there's, and a lot of them, including ourselves, continuous admixtures of antibiotic pressures, which would then sustain the presence of these multi-drug and even metal-resistant cassettes. So does this make a difference? This is really what we talked about today. Well, certainly I think we all can see that it could very well make a difference in understanding environmental fate and transformation. It could help us rethink toxicokinetics and some of the biomarkers we use to deduce the transfer of materials from outside the body into the body. Might add a dimension that could be of importance to understanding or predicting individual susceptibility. And finally, I think it does allow us to restore the importance of immunology and evolution to environmental health sciences, which is something that one of my students argued in a study in which she incorporated both the modeling, I won't go through this, of toxicology with the modeling for infectious disease, making the proposal that there could be an enrichment of both domains of science and research by thinking in a more collaborative, mutualistic way, perhaps. So I'd like to thank all the students who've worked on this, particularly Beth Feingold, who's here in italics because she wrote that paper, which appeared in EHP, a large number of colleagues from many disciplines, funding sources, and then the communities that we've worked with to study how this plays out in terms of human health risks. Thank you.
It was a wonderful summary, thank you. I just want to point out that I had introduced the horizontal gene transfer idea, but I was actually using Eric Alm's data from MIT, who approached that from a purely computational perspective. That was that first day. So there's early data coming out, kind of still collected, where apparently, we mentioned there's 100 trillion cells in the gut. There's tenfold more viruses in the gut than there are even cells. And those viruses apparently are breaking out as eukaryotic viruses and bacteriophage. There's also early studies showing that, early data coming out to show that there's actually a lot of circulating viruses even in our blood, and it's not clear what they're doing. So my question to you is, you didn't mention phage as a mechanism. Could you address that a little bit?
I would. I think, and again, this is from the somewhat restricted world of people who are concerned about antimicrobial resistance, but horizontal gene cassettes, plasmid conjugative transfer seems to be the dominant mode by which that is disseminated. That's not to say that we shouldn't think about these other things, but I think you and I would both agree we need to not be totally assured if the microbiome doesn't change a lot in terms of, say, functional or genotypic data based on 16S. Because actually what those bacteria are capable of doing, aside from their functions, may have changed a great deal.
Oh, indeed,
yeah.
Go ahead, John.
Yeah. I was just going to say, that was a wonderful presentation, and I understand in your last slide, your intent on that was to really focus it in on a toxicologically inclined institute, and
which would be you.
Which would be me, and I support that.
Yeah.
But I do think that we shouldn't shirk from another fundamental point that you're making, which is that it's not just a problem of interactions with metals, but it's a problem of antibiotic resistance and the enormous public health burden that puts on our society. And the question that you didn't hit head on, which is one that we've talked about and needs to be grappled with, is what is the contribution of these environmental and community acquired resistance genes to the terrible morbidity and mortality that we see in hospitals. We know that hospitals are factories for antibiotic resistance as well. There is this question of relative attributable risk to the extent that there is a significant influx, for certain pathogens from the community. There's enormous costs associated with that. And so, and that problem will not be solved with infectious disease thinking alone. It really requires environmental health thinking. And so I think that is a kind of a fifth component is expanding the concept of environmental agents to include these resistance genes and open that way of thinking.
And as you know, we've thought about that, and in trying to think about it specifically, we decided that our candidate organism would be methicillin-resistant Staph aureus. And we did that really for two reasons. There's a great deal known about the burden of disease and the costs related to MRSA. And the other is that over the past 10 or 12 years, as Vince undoubtedly knows, this has changed radically from being a nosocomial or healthcare-associated issue to what is now called community-associated. Unfortunately, that designation has really fallen like a thud in the world of infectious disease. And I don't know about your hospital, but I can tell you my colleagues across the street say, well, it's not our problem anymore, it's your problem. So we've spent a lot of time identifying settings where people get community-associated MRSA, but we really don't know where it comes from. And so with whole genome techniques that we're employing now in these studies, we can start to try and track where it's coming from. One candidate, and then try to figure out what would be the attributable risk of a particular activity, say livestock production, or anything else that's happening out there. And so we've studied, for example, did a case control study using a hospital which served a population in which there were a lot of pigs being grown, but also people living in towns. And we're now undertaking a study in North Carolina to look at the contribution of swine farming to the burden of MRSA infections, with a particular emphasis on children. You may know that in several countries in the European Union, Belgium, the Netherlands, and Germany, livestock-associated MRSA, defined genetically, is now contributing to 35 to 40% of the burden of diagnosed community-associated MRSA infections. So I think it's likely to be an example, not the only one, but perhaps a way that will help us, get our chops together to do other studies using the most advanced techniques.
Thank you. That's really wonderful. With that, I'd like to yield the floor to Helmut to bring your workshop.
Well, I'm not going to say much. I think Rita Shoney summed it up when she said this workshop was a revelation to those of us who don't work in this. And I think we all learned a tremendous amount, and the speakers were really given quite a difficult task of sort of thinking outside their regular way of expressing and thinking about this and really trying to relate it to environmental health. And I really applaud you for doing a great job making it understandable and relevant. I'd also like to thank all of the panelists and all of the liaisons for incredibly insightful and useful discussions. And I guess I'd just end by saying that it's important that we not end with this workshop, that you take this back to your agencies, spread the message, inform people how relevant this is going to be, and that we keep the momentum that we generate at this workshop. And with that, I'll say thank you to everyone and end it.
Let me also add my thanks and invite you all to some of the next workshops that we'll be having. There's another one coming up in July. Pathways and mixtures, which I think will be very good.